So in this video, I'll be going over the most important step in development, in my opinion, and that is a first cell fate decision in which a cell decides if it's going to be the cell of the embryo or it will make up the placenta. And so in a previous video, I went over the key steps in pre-implantation development. If you haven't watched that video, I'd go ahead and watch that first. And so this previous video did stress that around the marula stage in development, these cells are determining whether they're going to be the cells of the placenta or trophectoderm, or it will become the cell of the fetus or inner cell mast cells. And so in this video, I'm going to be going over mechanistically how this happens. Importantly, the first several divisions after fertilization are asymmetric, which means that one daughter cell will inherit the top portion of this original cell, inclusive of proteins which are typical of the top portion of a cell, or polar proteins such as PAR6, alpha PKC, and ERM proteins, which stand for ezrin, rhodixin, and myosin. And their sister cell will inherit proteins which are key to the bottom portion of the cell, which largely is these enriched actinomycin networks, which will ultimately lead to their internalization. And just to get a better understanding conceptually of how that happens, I want to take a step back and look at what the marula that is developing. And so here, following the same color schematic, we have in pale the cells which inherited the bottom apolar portion of the original cell, and then the cells which have inherited the polar upper portion of the original cells in red. And so these apolar cells have enriched actinomycin networks. And so what happens is actinomycin contracts and then generates high tension within the cell. And then due to Laplace's law, this high tension then causes this cell to internalize. And so therefore, what we get at the end of the day are these cells which have inherited the top or polar portion of the cells remaining on the outside due to lower tension generated, and the cells which have inherited the lower portion of the cell which contain these high enriched actinomycin networks generate high tension and are internalized. And so which cells at the end of the day are going to form the placenta, which ones are going to form the embryo? Are they going to be the outer cells or these inner cells? And to understand this, you first have to understand how a cell becomes a certain fate, because at the end of the day, right, each cell has the same genetic code, but they're just expressing different key genes, which are then being translated to make certain proteins which are specific to that cell, right? So for example, skin cells expressing just key genes which are important for making proteins of the skin, but they're suppressing via epigenetic markings genes which will then go and make proteins of the kidney or muscle or the liver or the brain, etc. And so if you can imagine, before a cell determines what fate it's going to be, it's expressing all different transcripts, all different genes. And so that is basically what a totipotent cell, and also you get into that in pathology and cancer, where, when you have, for example, a skin cell again expressing cells of the kidney, that's carcinogenic. But I digress, going back to the state of totipotency, where we have expression of all these different lineages. Around this 8-cell stage is the last stage in totipotency in humans. And then we get our first cell fate decision occurring around the marula stage. And so this is largely governed by a pathway called hippo signaling. And so what is this? Regular processes within the apolar inner cells. Typically, they cause activation of this kinase called LATs. And kinases, what they do is they phosphorylate. And so LATs will then phosphorylate this co-transcription factor called YAP. With the extra phosphorylation, it prevents YAP from going into the nucleus, and it stays in the cytoplasm. However, when you inhibit this process, then YAP can then enter the nucleus, and it binds to its co-transcription factor called TEAD, which is a key transcription factor for instigating the expression of key placental transcripts such as GATA3 and CDX2. And then what we see very commonly in development is this reciprocal inhibition of fates. And so when we have higher and higher increase in CDX2, which is a key placental transcript, then we have further and further inhibition of the transcript, which would generally make or govern the embryo lineage, which is largely OCT4. So now we have from going from a state, a totipotent state, which is expressing all these transcripts of all these different cell types, we now have this cell deciding that it's going to be largely specified as making the placenta. And these are the cells on the outside of the developing morula. And the cells which are in the middle, which are apolar, continue to activate hippo, and we don't get transcription of key placental transcripts. I guess the key question is, if these inner cells are maintaining their regular function and not being influenced to make placental lineage because of polar proteins, because they are apolar, then shouldn't they just stay totipotent? And so what scientists have found is that cells do express transcripts of the embryo, so OCT4, like slightly higher than those of the placenta. And so over time, what we get is in these inner cells, due to reciprocal inhibition and lack of hippo inhibition, we get OCT4 slowly increasing in the inner cells. And so those cells eventually will have fully dampened and inhibited the expression of the placental or alternative lineage, and will be solely expressing the transcripts of the embryo. So there we have it. The sister cells, which inherit the bottom portion of the original cell, will inherit these enriched actinomycin networks, and due to higher tension generated within those cells, will be internalized. Due to their apolar nature, they will continue and over time will be expressing transcripts of the embryo, and therefore these inner apolar cells will become the embryo. And then we have the other daughter cell, which inherits the polar proteins and less actinomycin network, and therefore generate lower tension and make their way to the outer shell of the morula. And then due to the polar proteins which they inherit, will inhibit the hippo pathway and will cause instigation of key transcripts of the placental lineage. Therefore, these outer polar cells will then become the cells of the placenta.